This is where we left off with lysosomes, and also we didn't really talk much about food vacuoles, but we went from the Golgi apparatus, and remember, Golgi apparatus will create these lysosomes. And one of the reasons why they are membrane bound is because they have enzymes in them that can digest or break apart uh, larger molecules into smaller. And generally, once a larger molecule is broken into a smaller one, it can no longer perform the function it was doing before. And the only reason I say this is that's how a lysosome helps protect the cell and protect things that are in the cell that are potentially toxic. But the enzymes that are in the lysosome operate in a certain pH, and it tends to be more acidic than the pH of the cell proper. So the cell plasma membrane, or cell cytoplasm. And so it needs to be walled off. If they happen to break open, that pH can be neutralized. If a lot of them break open, as it can happen, that then will cause the cell to die. And it is one way that the body takes care of old cells that need to die to be replaced by newer cells, is having multiple of these lysosomes break apart. In this process of cell recycling called autophagy, so AUTOPHAGY, is when multiple lysosomes break open, and that changes the pH of that cell and causes it to die, unfortunately. This idea of vacuoles is a very, very useful I guess catch-all, you might say, in terms of when we talk about just digestive or compartments in the cell, not necessarily digestive compartments in the cell. But vacuoles can be very useful for many reasons. You can have a food vacuole, and on the image on the right, you have, if you look at the plasma membrane, which is the gray portion, and drop down to about the middle of it, you have a food vacuole, a piece of food that actually is being brought into the cell in a process called phagocytosis. And what will happen is that food vacuole will then become one with a lysosome, and that lysosome will allow that food to be broken apart into what the cell can use, and then released into the cell, so the cell has the parts it needs to run whatever energy machine it works on, whether it works on the mitochondria or works on chloroplast or mitochondria or whatever it works on. And then you also have here an image of the lysosome being created by the Golgi apparatus, and lysosome actually joining with a something that needs to be neutralized, this autophagy, and digesting it. Digesting it because it has enzymes in it that can break it apart. Other things that vacuoles can be, they can be food vacuoles. If it's a smaller cell, it can be a contractile vacuole. What that will do is, if you have with cells that live in a fluid environment and are constantly taking in fluids, a contractile vacuole can actually be formed to actually really shunt large amounts of fluids out of the cell. It can also be used to bring large amounts of fluids into the cell to create a larger cell without the need to make more cell membrane. So for a short term, you can have a much larger cell with a vacuole in it filled with water, and then without having to go through the process of making more cell membrane. Just an overview of this endomembrane system. That's what we've been talking about for a while. First, you have the nucleus, and the nucleus is the nuclear envelope is where you have the interface between the nuclear envelope and the endoplasmic reticulum. Remember, you have ribosomes on the nuclear envelope as well as ribosomes on the endoplasmic reticulum, which at that time we call rough ER. And that's the surface of the ribosomes on the rough ER are going to create proteins. In this image, it's called polypeptides, so multiple protein units. They're kept in the lumen of the ER. They then go to the smooth ER. Smooth ER packages the protein in vesicles, sends it to the cis face of the Golgi apparatus, then merges with a vesicle and it transports the protein, modifies it, ready for delivery. So it comes in, the Golgi looks at it and says, okay, yeah, it's ready. And then in delivery, it'll pinch off from the transface of the Golgi apparatus into a transport vesicle and taken outside the cell. Generally, those things that are made and need to be made and used just inside the cell are going to go to the free ribosomes. Remember that. That's the endomembranous system. A little bit more about vacuoles just as an image, particularly in plants or fungal cells. As I said, they have food vacuoles for phagocytosis, for eating, just like we think about the food. Contractile vacuoles for to pump out excess water that accumulates or comes into the cell just in the course of them actually taking in food and taking in nutrients. And then a central vacuole that can hold not only water but also compounds that are needed, so nutrient compounds that are needed for the cell. And that's why this central vacuole, as I said, has the ability to change the size of the individual cell if it's filled up or smaller. And that's why I have this picture here about a full vacuole. That's when your plant is perky and upright. And when your plant is sagging and the leaves are drooping, that's when you know it's time to water it so that it takes up the water, and then you have full vacuoles again. So next time you water your plant, you can say that you're filling its vacuoles. Moving on to another, these are membrane-bound organelles. So let me say that more clearly. Membrane-bound organelles, but they are not part of that endomembranous system. So these are the types of organelles that are used for energy. If it's a plant, it's mitochondria. If it's an animal, it's mitochondria. If it's a plant, it's a chloroplast. So if it's an animal, it's a mitochondria, and that's the image that you have on the left. And if it's if it's a plant, it's a chloroplast, and that's the green thing. And that's what you have on the right. I think I just said that wrong. Animal on the right, mitochondria. Oh, I should just start over again. I'm sorry. Mitochondria is on the left, chloroplasts on the right as you're looking at it. They convert energy to ATP. And what you have here is a design of how did these things enter the cell, because they're unique items themselves. And there's this process or this theory that there was a time when they were taken in. It's called the endosymbiont theory, that a mitochondria was taken into a cell, chloroplast was taken into a cell because for protection, that cell would actually be able to protect it, and it just stayed there. We evolved with the mitochondria in there. Mitochondria actually has different DNA than we have. You do inherit your DNA, but it is different than the DNA that you find in the nucleus. And then, these images are very interesting, but they don't necessarily tell you everything about the mitochondria or the chloroplast. Mitochondria, animal cell; chloroplast, plant cell. cellular respiration is the process we talk about with each of them. They just have a different pathway they take for cellular respiration. With mitochondria, it's a product from glucose that goes into a cycle we call the citric acid or Krebs cycle. And with cellular respiration in plants, the energy comes from light, is captured by those chloroplasts, and then that energy is taken into the Krebs cycle to produce ATP. Here's the mitochondria itself. It's a unique design, and one of the reasons why I put the formula on there, carbohydrate plus oxygen equal carbon dioxide, water, and energy, is because I think the more times you see it, the more times you have the opportunity to get it in your head. So what happens with regard to our chosen form of energy is a carbohydrate. Now understand, it's not the only form of energy that we can use in the mitochondrial cycle, but the chosen form is carbohydrate in the presence of oxygen. So this is an aerobic reaction, and the formula, the transaction takes place, and what happens in the end is we have energy, which is what we want. We have carbon dioxide, which is a byproduct, and water. Mitochondria is the site of cellular respiration, and the end product that we are trying to get to is ATP. Chloroplasts in plant and algae for photosynthesis. We'll talk a little bit about photosynthesis in another chapter, and actually more about the process of energy production in the mitochondria. Mostly what you're just doing here is learning the design of the mitochondria and how the design, the structure, leads and function are interrelated. They contain their own DNA, so mitochondria contains its own DNA, and mitochondria are found in almost all eukaryotic cells. They have a smooth outer membrane, an inner folded membrane called a cristae. So similar to your Golgi apparatus in this folded membrane, but certainly very, very different, because you have you have more enzymes, and certainly different enzymes in the mitochondria. The inner membrane has two compartments. You have a space and a matrix. So you have an area that is creating the space and the space. The cellular respiration takes place, so the the metabolic steps of cellular respiration, the chemical reaction, you might think of it that way, takes place in the mitochondrial matrix. So you have this area that is uniquely designed basically to withstand and store and process high energy reactions. In the cristae, you have enzymes, so enzymes that are unique to synthesize ATP. So smooth outer membrane in the mitochondria. Inner membrane has two compartments where you have a space and then a matrix. In that matrix are enzymes that are very, very important for ATP synthesis. Chloroplasts, on the other hand, also have a double membrane. They have a fluid-filled stroma with enzymes for carbohydrate synthesis. Plants actually make the carbohydrates that we use. Inner membrane, where you have thylakoids and grana, contain chlorophyll that can absorb solar energy. So this chlorophyll has a special adaptation to actually capture the energy from the sun and then use that energy to run its own processes. Chloroplasts are part of the plastid organelles. We are going to pick this up on the next one.